Okay, so today I would like to talk a little bit about what the Stein lab does and why we do it. I'm a neuroscientist. I'm generally interested in how the brain functions. I would like to understand how neurons communicate, how they produce behavior, how we respond to sensory inputs, and how the brain comes up with adequate responses to its environment. So we study a small part of the crustacean nervous system. It may sound odd, but there are a couple of advantages. So if you want to understand how the brain works, you're faced with a couple of problems. The biggest problem is that the brain is incredibly complex. There are many different brain structures, but even if you look within those brain structures, they are extremely complicated, they're extremely complex. There are a lot of nerve cells in there. So to understand the very basics of it, we actually have to look for simpler systems, or let's put it this way, at least systems that we can handle in terms of numbers and sizes. So the crap that we use for our studies, that particular part of the nervous system is called the stomatogastric nervous system. It is special because in that little part that we look at, we have only 26 neurons. So 26 neurons, that sounds like a number we can handle and be able to understand. At the same time, those 26 neurons do something very important. Those 26 neurons are part of what we call a central pattern generator, and a central pattern generator is a neuronal circuit that produces rhythmic output without having any rhythmic input. So that's a very basic function of the nervous system, because many of your everyday behaviors are based on rhythmic activity. So for example, breathing, right, is a rhythmic thing. And so you have neurons, central pattern generators in your brainstem that control the rhythmic behavior that we call breathing. Or, locomotion. In most animals, including humans, there is a central pattern generator that drives locomotion. Now, in order to understand how these things function, and in particular when they malfunction, so if they don't work anymore, to understand why they don't work, we need first to understand, how they work. So we have to start small, and so a central pattern generator that has only 26 neurons, is a prime system to study how rhythmic activity is produced. Now, the system has been studied for quite a long time, more than 40 years, more than 4 decades. And you might wonder, okay, what do these people do? 26 neurons, 40 years, probably, many dozens of labs who worked on that. So what do these people do? Well, the first thing that was done was essentially to establish how are the neurons connected. Now, this is nice, and this was done within a few years, right? The good thing about this is 26 neurons, you can use small electrodes, you can put them into those neurons, because those neurons also have the advantage that they are large, and you can establish the connectivity between those neurons. Now, people quickly came up with what we call the connectome today, essentially how neurons are connected. And here's the interesting thing. Initially, 30, 40 years ago, people thought once we know the connectivity, we will know how a particular system works. And in fact, a lot of people still believe that today. It's a very common thing to say, we map all the connections in the brain and then we know how it works. Unfortunately, that's not the case. With those connections, and there are quite a few, when you think about it, it's 26 neurons, and if everybody is connected to everybody, we have a lot of connections. In any case, those connections are not always functional in each condition. So, meaning, say you're in a particular sensory condition, you smell something, you see something, you hear something. Some of these conditions get blocked, others get activated. And that changes the behavior of the animal or the human, and it changes actually the structure, not physically, but the functional structure of the central pattern generator. So now that comes with a couple of problems, because now all of a sudden the connections that we have are just the basic, right? The basics. They are just what we use to describe in general how that thing might work, but now we have to figure out in different conditions when are certain connections activated, when are certain connections blocked. And this made the system actually much more interesting. What is this person doing? This made the system much more interesting because, you know, once we had all the connectivity, we actually were able to address the bigger elephant in the room, which is, you know, how do they change over time and how do they change in different conditions. Now, the system that we work on in particular has a way of modifying the structure in terms of the strength of the connections and how they function. In that case, that particular influence that changes the connections is called neuromodulation. Those are substances that are released into the blood, for example, or, you know, released from other nerve cells. For example, dopamine or serotonin. I'm sure you have heard of those. Those are called neuromodulators because they do change how neurons respond to incoming information. They also change how neurons talk to one another. In other words, the connections, how they are changed. So it was found that in this system there are more neuromodulators that modify the central pattern generator than there are neurons themselves. So for those 26 neurons, by now we know of more than 75 different neuromodulators, different chemicals that alter the functioning of that circuit. Now, you might think, okay, this could be something that's specific to, you know, the crab. You're looking at a crab after all. But no, that's not the case. In fact, many, many years after it was found, you know, in systems like the one that we work with, studies in the vertebrate, or mammalian, I should say, breathing system have found that there is an equal amount of neuromodulators and that the system is equally complex in terms of how it changes in different conditions. It's just much more difficult to understand what it does, which is why it took a lot longer to figure that out in this other system. Their ideas were based on the studies in the system that we work on in the crustacean nervous system, for example, but also other smaller circuits. So, now these days, what we do is we're interested in how these neuromodulators are controlled. Now, how are they released? In what conditions are they released? How do they act together? What happens, for example, if dopamine and serotonin are there at the same time? Those are difficult things to study for the same reasons as I mentioned before. In complex nervous systems there will be complex interactions, so we need to start easy or small. Now, how do neuromodulators interact? Well, it's an important question also because when we think of treatments for, say, Alzheimer or Parkinson's, where we deal with neuromodulators, right, like dopamine, and their antagonists, or depression, serotonin antagonists, and so forth, we need to understand how the different drugs interact. And so this is something that we are interested in the lab. We do this using a small circuit in the stomatogastric nervous system of the crab. And I think it's quite valid to study this system because of all of its advantages. If you're interested in the specifics, I recommend looking at my other videos. or you can always email us, or you can schedule a visit and come see us in action. We're always happy to show visitors our research, and I would be happy to explain in more detail to you what we do. Thank you.